why don't we blow the system up? I mean, obviously we can't just turn off the spigot on the system we have and then say, hey, everyone in the world should get this new vaccine we haven't given to anyone yet. But there must be some way that we grow vaccines mostly in eggs, the way we did in 1947.
In order to make the transition from getting out of the tried and true egg growing, which we know gives us results that can be, beneficial. I mean, we've done well with that, to something that has to be much better. You have to prove that this works, and then you've got to go through all of the clinical trials, phase 1s, phase 2s, phase 3, and then show that this particular product is going to be good over a period of years. That alone, if it works perfectly, is going to take a decade.
There might be a need or even an urgent call for an entity of excitement out there that's completely disruptive, that's not beholden to bureaucratic strings and processes.
So we really do have a problem of how the world perceives influenza, and it's going to be very difficult to change that unless you do it from within and say, I don't care what your perception is, we're going to address the problem in a disruptive way and in an iterative way, because you do need both.
But it is not too crazy to think that an outbreak of a novel avian virus could occur in China somewhere. We could get the RNA sequence from that, beam it to a number of regional centers, if not local, if not even in your home at some point, and print those vaccines on a patch and self-administer.
Given, as you heard from the introduction, that I have been around for a while and have had the opportunity and the privilege and the pleasure of serving in 5 administrations, I thought I would bring that perspective to the topic today, is the issue of pandemic preparedness. And if there's one message that I want to leave with you today, based on my experience, and you'll see that in a moment, is that there is no question that there will be a challenge to the coming administration in the arena. There will be a challenge to the coming administration in the arena of infectious diseases, both chronic infectious diseases in the sense of already ongoing disease, and we have certainly a large burden of that. But also there will be a surprise outbreak. But also there will be a surprise outbreak. But also there will be a surprise outbreak. And I hope by the end of my relatively short presentation, you will understand why history, the history of the last 32 years that I've been the director of NIAID, will tell the next administration that there's no doubt in anyone's mind that they will be faced with the challenges that their predecessors were faced with.
Then we started looking, where did they come from? And we went out to southern China and did surveillance of bats across southern China. And we've now found, after 6 or 7 years of doing this, over 100 new SARS-related coronaviruses, very close to SARS. Some of them get into human cells in the lab. Some of them can cause SARS disease in humanized mouse models and are treatable with therapeutic monoclonals, and you can't vaccinate against them with the vaccine. So these are a clear and present danger. Yeah. We've even found people with antibodies in Yunnan to SARS-related coronaviruses. So there's human exposure. Right. We're now doing surveillance. We're just beginning another 5 years of work to look at cohorts in southern China to say, well, how frequent does this spillover happen, and is it associated with disease? Because I expect, just like Nipah in Bangladesh, there are dozens and dozens of small spillovers going on the planet at any one time, which we just never see.
Sure. Yeah, they never amplify. There are a few cases, and they're just absorbed in the illness, the overall illness of the population, right?
Now, you could say, so who cares? And that's one argument. But our strategy is any one of those could become pandemic. There's a lot of stochasticity in what happens then. Yeah. So if we look at all of them, understand patterns, try and reduce the number of spillover events, we've got,
But if you're saying these are diverse coronaviruses, and you can't vaccinate against them. There are no antivirals. What do we do?
Well, so I think that coronaviruses are pretty good. I mean, you're a virologist, you know all this stuff, but you can manipulate them in the lab pretty easily. It's just spike protein drives a lot of what happens with the coronavirus, zoonotic risk. So you can get the sequence, you can build the protein, and we work with Ralph Barrick at UNC to do this. Insert it into a backbone of another virus, and do some work in the lab. So you can get more predictive when you find a sequence. You've got this diversity. Now, the logical progression for vaccines is if you're going to develop a vaccine for SARS, people are going to use pandemic SARS as
sure.
But let's try and insert some of these other related and get a better vaccine.
And I guess also knowledge of what's there. If you see something emerging, it give it a head start on making a vaccine or a therapeutic.
That's true, and better knowledge of where they are as well, so that you can put your money into the clinics that matter.